Alrighty then, Anna here. We are now going to do part 1 of the muscular system. And I've pulled together histology slides from both the beginning and the end of the PowerPoint and kind of organized them into one space. So let's get to it. Alright, so this is just a note slide. You should know all of these components: excitability, contractility, elasticity, extensibility. These are not the same thing as stretching. Okay, so remember, I've talked about that before. Be careful with how you use language. All right, let's go ahead and move on to the next slide. All right, you need to organize your information so that you have a way to quickly study the traits for these types of muscle tissues. This is a beginning for a table to help you organize that information, okay? So location, characteristics, control, okay? You're going to want to add information to whatever table you build later on using the tables that we have before. So you need to take all of this and consolidate it and shorten it, okay? And you will remember it better if you do it, okay? All right, next slide. All right, so now we're going to start by looking at the different types of tissue. And so we're looking at cardiac muscle tissue. And there are several things that help you with identifying these structures. The first thing that you want to memorize is that they have a branching pattern. The next thing you want to memorize are the presence of intercalated discs. Okay? And then the least important thing is striations. The striations are a reflection of the way the sarcomeres are structured, the same in skeletal muscle and cardiac muscle. However, whether the striations look distinctive or faint or whatever depends on the quality of the staining when they made the slide. So all you can remember is that both cardiac and skeletal muscle have striations. Okay, but you can't say whether one is strong or not in the other because it just depends on the thing. Okay, now I'm actually going to reduce this for a minute, and I want to draw out what to me looks like a cardiac cell. Okay, that's what a cardiac cell looks like to me. So that's a cardiac cell. Okay, now I want to show that next to a skeletal cell. So I'm going to draw a single skeletal cell right here. Now notice what I've done. Skeletal is really long. Cardiac is much shorter. Okay? Skeletal muscle cells usually are so long they do not all fit on the slide. All right? But I can get a lot of cardiac cells on a slide. Then you've got smooth. Okay? And smooth muscle is going to be kind of like that. So that's a cell. That's another cell right there. There's another cell there. There's another cell there. There's another cell there. So each one of those is a single cell. Okay? So smooth muscle cells are really tiny in comparison to cardiac or skeletal. Okay? So skeletal is long and linear. Smooth muscle is tiny and spindle shaped. Whereas cardiac is short but seeable, and it branches. Okay, now let's blow this back up so that we can see it. Now, I want you to pretend that this used to be a river, and then I took a big rock and I dropped it in the middle. Okay? Remember that cardiac cells and skeletal muscle cells all have myofibrils that are running down it, okay? So they're all going like this, okay? So these are representing the myofibrils. What's going to happen is that they're going to be going like this, and they're going to be doing just fine, okay? But then they're going to hit this rock, and they've got to make a choice. They've either got to go this way or they've got to go this way, okay? This rock is a clump of mitochondria, all right? Cardiac cells are aerobic, which means they have to have a constant supply of oxygen for the mitochondria to make ATP energy, okay? So cardiac muscle cells have a plethora, a ton of mitochondria. So many that it causes the myofibrils to go into different directions, okay? Now I'm gonna actually kind of redraw this so that's a little less messy down here. And then I'm gonna do that. And then I'm gonna show it next to another muscle cell. We're gonna do that. And then I've got another one over here, and it's going to do something like that, blah, 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 et cetera, et cetera. This is called the intercalated disc. And in your intercalated disc, you have two things, gap junctions and desmosomes. And you need to memorize this. Gap junctions allow charged ions to pass between the cells, all right? This creates a functional syncytium, and we're going to focus on this part of the word, syncytium or sync. It allows the cells to sync up so that they all contract as a single unit. Desmosomes are, remember I described them as shoelaces that tie together the cells, okay? These cells, when they contract towards their center, okay? That means there's a tremendous tension at that intercalated disc. So the desmosome's job is to prevent these cells from ripping at that point. Now, desmosomes are full of a protein called keratin, and we saw this in the integumentary system. Now think back to the stratum granulosum. How did it stain? I want you to visualize it in your mind's eye. What made the stratum granulosum look different? All right, you got it in your mind's eye? Hopefully you're thinking those grains or spots because there's extra keratin in the granulosum. So what does keratin do? It's really good at absorbing stain. Because of that, intercalated discs tend to stain a darker color. Now let's look at this drawing. So there is an intercalated disc. There is an intercalated disc. Here I can see this branching pattern, okay? I tend to have just one nucleus because these are short cells and they don't need a bunch of them, all right, because they're all pretty close together, all right? All right, let's move to the next slide. Wow, that did not import well. Anyway, this is one of the slides that from the A&P lab that we have on campus. This is my black stain slide that I really like. And the reason I like it is because it's really easy to see the intercalated discs. It can be difficult interpreting your muscle cells. So if you see an intercalated disc, it is a dead ringer for cardiac tissue, okay? It is harder to see it, but you can sort of get an appreciation for this branching pattern here. So you've got this and you've got that. Let me see if I've got a better one over here. It's not. Everything looks wonky because of the way it imported. So I can kind of see cells here, and then it's kind of going like right here. It's not super straight and linear the way skeletal is. But I also have these dark circles, and these are the nuclei. All right. It looks like there's a lot, but there's actually only one per cell. The reason it looks like a lot is because this is not a great cut, and they've got more than one cell stacked on top of each other, so it wasn't thin enough. All right, let's look at the next slide. All right, this is the model slide, and I have no idea why that arrow is showing up in the background like that. Actually, it kind of looks like it turned these upside down when it converted to a PDF. This is just kind of strange. Anyway, what I like about this model is you can see certain things. So this is that intercalated disc, okay? I can see really nicely the branching pattern right here, okay? So this is cell number 1, and this is cell number 2, okay? I also like that you can see the T tubules right here. You can see the sarcoplasmic reticulum. I can see the striations of the sarcomeres. I can see the mitochondria here. So lots and lots. I mean, look at how many mitochondria are in there. There's tons. And then just one little dinky nucleus right there. Okay? So I like the model because I think it's a little bit easier to see some of those structures. All right, let's look at the next slide. All right, so smooth muscle is involuntary, like cardiac. Oh, what word am I writing there? Okay. The thing with smooth is my number one clue for what I'm looking at is context. All right. Smooth muscle is going to be in and around all your mucosas, all your mucous membranes. So all the tubes and structures of your body are going to have these. Now, this is kind of a typical magnification. So this is probably about 400x. This is also at 400x. And what you're going to see is it's very difficult to see the individual cells. So they're taking this and they're blowing it up here. And you can see you've got this very thin spindle shape with a nucleus in the middle. And then the next cell would be kind of like this, and it'd have a little like that, and it'd be like this, and kind of like that, with the nucleus in the middle like that. And they're all kind of lined up like this, okay, with a little nucleus in the center. They're very, very thin and very, very short. And it's really hard to see them. So if we look over here at this picture, you can't really see the individual cells. What I can see are the nuclei all going kind of in a linear direction. I've got the lumen of a tube, therefore I know for a fact that this is a mucosa. So I'm seeing something that stains pink. It's got purple lines. To me, this always looks like a river of fish swimming in a circle. And so they're all going together. All right? Now, this is also smooth muscle here, but it's been cut So these are the heads of the fish sticking up straight because they stopped swimming. Okay? Like you've cut their heads off. All right, so context. Context is the key usually for identifying your smooth muscles. All right, let's look at the next cell or slide. So, whoops. All right, here is a model. So this is a cell. So this is cell number 1. We've got cell number 2. We've got cell number 3, cell number 4. So they're all stacked up here together. So you get an idea of how it's got this spindle shape right here. You can see a nucleus right there. Okay. Now what you're going to appreciate is that it does not have the same structure of myofibrils that skeletal muscle and cardiac muscle does. All right. I don't need you to get bogged down in that too much. It does have contractile organelles, but everything is arranged a little bit more loosely, and the sarcoplasmic reticulum is arranged differently, and we'll look at that again in a few minutes. All right, so again, I don't know why when this converted to PDF, it brought over these arrows, so you're just going to have to ignore them. So this is my notes slide. I need you to know basically that you've got single unit, which means they all function together to form a synthium like in cardiac, okay? They are connected by gap junctions, and basically it allows them to all work together, all right? And then you have multi-unit smooth muscles, which we're not going to spend a whole lot of time on. All I basically want you to know is that the arrangement is a little bit differently, whereas with single unit is really about the synthium and the gap junctions working together. All right. Multi-unit is going to cluster cells which are controlled by then a neural stimulus. So it's actually a little bit more similar to skeletal muscles, neuromuscular junction. And it's going to be into some of these larger structures like arteries and sphincters and stuff like that. Okay, and that's about it. All right, here we're going to look at a single unit, but it's like the most classic single unit smooth muscle. All right, so we're actually looking at intestines right here. And so this is my lumen. This right here is my mucosa. And so I know from context that I'm dealing with one layer of smooth muscle there and another layer of smooth muscle, okay? This is what I was describing as the fish all swimming in one direction. So these are my fish swimming in the river. Now, so if I've got an intestine like this, I can look at muscle running along linearly, or I can have muscle that coils around it, okay? And when I cut through here to make a slide, the linear is going to be this. Whoops, need a different color. And then the circular is going to be this, okay? So that you can see the pattern of the cells a little bit differently. So this is where I describe the fish getting their heads cut off, all right, because they're swimming the length of the tube, okay? So this is longitudinal. Okay, and the different types of arrangements allow you to do different things. So in AMP2, we'll talk about segmentation versus peristalsis, both controlled by smooth muscle, but the longitudinal layer allows a wave-like contraction for peristalsis, whereas segmentation with circular fibers does a back and forth mixing of whatever it is you've got. Okay, all right, let's look at the next slide. All right, so now we're going to look at skeletal muscle. And what you see is a slide. Now, this goes in this direction and that direction, okay? In here is a section of cell. That's a cell. That's a cell, okay? In here, this is where you're going to find the layers of endomysium between the cells, okay? So that's in there. The nuclei get squished off the sides, but I can see I've got 1, 2, 3 in just this little segment. So there are multiple nuclei in every one of these cells. Okay? All right, let's look at the next slide. All right, so this is the neuromuscular junction, which we're going to spend more time on later. But what I want to show you is right here, this particular model. So we're going to blow this up and we're going to kind of make it center stage. And I'm going to label it. So this right here, actually I'm going to get a different color that we could see better. Of course I picked black. That's your sarcolemma. Okay? Whereas that is endomysium. And we'll talk more about that in a bit. All right? In here, each one of these things, actually, let me redo how I'm portraying that. Let's go with green. So each one of these little things, every single one of those is a myofibril. Okay? And then the myofibril is the organelle that does contraction. Okay? So you've got your sarcolemma here. You've got the myofibrils here. And then here you see a nucleus. All right? And I've got another nucleus here. And do I have another one that I can see easily? I'm not seeing it. All right, and then this is going to be the neuromuscular junction, which we'll talk more about later. Okay? All right, let's look at the next slide. All right, so now we're looking at the photomicrograph of skeletal tissue. And what you can see are good striations. Change color. Good striations. Okay? But also it is long and linear. It's going like this. I don't see breaks. I don't see branches. It's very smooth. It's very linear. I don't see intercalated discs. I do see quite a few nuclei. Okay, so that's what I'm looking for when I'm looking for skeletal muscle. Let's look at another picture. All right, here we've got a pink staining slide. All right, and again, it is long and linear. So it's linear. I've got striations, okay? And this would be the longitudinal area, and this would be longitudinal, this would be longitudinal. Over here, this would be a transverse cut, okay? And then, of course, lots and lots of nuclei. I don't see intercalated discs. I don't see branching. I have striations, okay? So it's not smooth muscle because I've got striations. It's not cardiac because there's no intercalated discs and there's no branching. All right, next slide. All right, so now we've put them all together. So here is cardiac. There's my intercalated disc. Over here is skeletal. No intercalated discs, long and linear. It's difficult to tell, but both of these have striations. Okay, now up here is my smooth, and I'm going to use context, the fact that this is a mucosa lining a blood vessel, okay, right here. So this is my mucosa. And so here is the smooth muscle, and here is the smooth muscle as well, but it going in different directions, okay? So you need to find a few tricks that help you distinguish these different types of muscle, okay? Let's move on. All right, so from another book, I borrowed a bunch of tables that summarize and do basically a compare and contrast of skeletal, cardiac, and smooth muscle. I am not going to read these to you, but these are the traits that you're going to use for the EC muscle types trait quizzes. Let's kind of flip through them. I'll say a few things here and there as I'm inspired. All right, so I showed you a table at the beginning of one that I was starting to make. So you could modify that and start kind of putting these things in there. What you're going to notice is on the EC quizzes for this topic, they come right off of these. I mean, just like almost pretty close, okay? So have a column for striations. Smooth would be no, skeletal yes, cardiac would be yes. Sarcomeres, no, yes, yes, okay? Thick filaments, the troponin complex, arrangement of fibers, sarcoplasmic reticulum, less developed versus more developed, the way the neuromuscular junction works, calcium gap junctions, so forth. Okay, now here, maybe I should pull this up earlier so that we can show this off with smooth muscle. We just do not give enough time to smooth muscle in A&P1, and even though it's more important for A&P2, we also do not give it enough time in that course for you to really understand it. So you're going to need to kind of remember that we did this in A&P1 and then go back and refresh your memory when you get to A&P2. So when you look at the structure, so you've got those spindle-shaped cells that are all kind of next to each other, all right? They're going to have these gap junctions between the cells so that things can move between them. Now, these varicosities, so when you ever see the word varicosity, you're looking at a web. It's like a web of nerve endings or a web of blood vessels. But basically, I have autonomic, so these are involuntary nerve fibers that are going to come up to this smooth muscle. And they've got these little bulgy spots here. And all of these can trigger depolarizations in these areas and by releasing these neurotransmitters. And because of the gap junctions, then these will all sync up and act as a synthium so that all of these cells can contract as a single unit. Okay? Now, if we start to go inside to look at these, so what they're showing in this picture are the gap junctions here and here. Instead of the whole sarcomere structure, we've got these things called dense bodies instead, okay? And then the filaments, those myofilaments, the myosin and the actin, they're there, but they're arranged in this kind of web-like pattern. And then they're going to work with the dense bodies in order to do the sliding filament mechanisms in this kind of webby-like direction. Have you ever seen the toy? It's the Chinese finger, what are they called? Chinese finger trap or puzzle. You stick your finger onto either end, and if you pull, it traps your finger to get them loose. You have to put it together. I kind of visualize smooth muscle working like that, okay? And the way the sliding filament mechanism pulls things towards the center in this kind of webby-like structure. All right, let's look at the next slide. All right, smooth muscle is weird, okay? What I want you to notice is that there is a sliding filament mechanism with actin and myosin. It is triggered by calcium. ATP energy is required for cross bridges to detach, okay? When calcium levels drop, that's when these things go into relaxation mode, okay? Now, so these are the things that are the same, okay? But there are some functional differences to smooth muscle that alters how it's going to be shaped. First of all, smooth muscle is going to be able to contract 30 times longer and hold it for longer, but it only needs 1% of the energy, okay? This is a fairly slow process, but it's a very sustained contraction that doesn't require a lot of energy. This is very efficient, okay? Now, in terms of the neurons, with skeletal muscle, all connect up to nerves. With smooth muscle, you're going to be using some autonomic and some nerves and some other things, okay? Smooth muscle cells, I can make more of them, or I can make the cells fatter, all right, thicker in diameter. Over here, I can only make the cells bigger in diameter. They don't go through mitosis. They don't split, all right? Let's look at the next slide. All right, this is a nice picture because it's got skeletal, cardiac, and smooth. Gives you some basic locations, and then it gives you the photomicrograph of each, okay? And then we're going to keep going on. What I like about this one is it shows you the way the cells are put together. So this is our classic one that we looked at in class. Cardiac is very, very similar, but we have bigger webs of endomysium, thicker endomysium between the cells. That also contributes to that branching pattern. Smooth muscle also has endomysium, okay? Now over here, presence of myofibrils composed of sarcomeres. Yes, but myofibrils are of irregular thickness, and this has partially to do for those mitochondria again, okay? No for smooth, all right? It's going to do that web instead with dense bodies anchoring the actin filaments, all right? This one is very, very interesting right here. I have 2 T-tubules in each sarcomere, okay? I have them in cardiac, but I only get 1 per sarcomere in cardiac, okay? That makes sense because these are shorter cells, okay? So pay attention to that. And then no T-tubules or etc. in the smooth muscle. All right, looking at this one, pay attention to this. So elaborate sarcoplasmic reticulum. We've got it in cardiac, but it's less distinctive, okay? In smooth muscle, we've got some sarcoplasmic reticulum, but it's not a whole bunch of it, okay? Gap junctions, none in skeletal. Yes in cardiac, yes in smooth. Not all of it, but in much of it. The neuromuscular junction, yes. No, except if you're in multi, in which case you do. Voluntary, involuntary, involuntary. All right, here we are on our last slide, and this is interesting. It's showing you sarcoplasmic reticulum, all right, is where you get your calcium. So this is where you're getting calcium. For cardiac, you get the calcium stored in the sarcoplasmic reticulum, but you also get a higher flood of it coming in through the cells. This is going to be important in A and P2 when you start studying the physiology of cardiac tissue, but also pathophysiology when you do the cardiac unit. All right, in smooth muscle, you're getting it from the same place as cardiac. So that's a difference. With skeletal, you are not getting calcium from the extracellular fluid. All right, where do you regulate the calcium? Around the troponin, but we don't have this same mechanism in smooth muscle. So you got these little doohickeys called calmodulin and the cytosol that regulate it. All right, and I won't ask you to explain it, but just be aware of it. All right, cardiac and smooth will have pacemaker cells, whereas skeletal will not. Skeletal only excites, but cardiac and smooth can excite or inhibit. All right, the structure of those myofibrils allows skeletal muscle to contract really quickly if it needs to. You lose some of these structures up here, the 1T tubule, all of that kind of stuff, and that slows down the ability of that cell to contract. Smooth muscle chucks a bunch of those structures, and it is very slow. It's strong, it's powerful. It can hold that contraction forever using very little energy, but it is very slow to get started. So when you are trying to run away from a coyote, you need the mechanisms to make skeletal muscle move faster, okay? And then you can do aerobic or anaerobic, and then these are basically aerobic. All right, so the EC Essential Concept Muscle Type Traits quiz are coming from these tables. So you need to study these tables in order to do that quiz, okay? All right, so that was the end of this part, and I'm going to go ahead and stop the recording.